The overall goal of the following experiment is to understand differences in brain physiology associated with recognition of pictures versus words in patients with amnestic mild cognitive impairment and Alzheimer's disease. This is achieved by using EEG to measure scalp electrical activity while patients with Alzheimer's disease are making recognition memory decisions for pictures and words. To accomplish this, the subject should be prepared for electrode placement and EEG setup, and scalp spatial localization data should be acquired using a special 3D digitizer. Next, the behavioral memory task should be run while raw EEG data are recorded. Then, in order to examine event-related potentials related to memory for pictures and words, the raw EEG data must be post-processed and averaged into ERP waveforms for each condition. These averaged ERPs can then be statistically compared across conditions. The results obtained in this study show that memorial familiarity or conceptual processing of pictures, but not words, appears to remain intact for patients with Alzheimer's disease compared to healthy older adults. This is accomplished by examining the early frontal ERP old-new effect, which occurs from approximately 300 to 500 milliseconds post-stimulus onset.
Hi, I'm Brandon Alley, an assistant professor in the Department of Neurology and director of the Memory Disorders Research Lab at Vanderbilt University.
Hi, I'm Erin Hussey, and I'm the lab manager for the Memory Disorders Research Lab.
Today we're going to be showing you some experimental methodology to understand brain physiology in patients with Alzheimer's disease. This method can help us answer key questions such as what aspects of memory remain intact in these patients. And Erin will be showing us the methodology today.